Hey all, welcome to ShareTreck, where science meets investing. I'm your host Raj. Today, we are going to explore a company called BEAM-302, the company leading the charge on base editing, which is a next-gen gene editing platform, which I think could potentially go beyond CRISPR as we know it. I value your time, so I'll keep this video crisp and to the point. If you have any query or any clarification that you want about this video, please drop them in the comment section. And as you know, I respond to each and every comment. And that said, let's get started. I would like to start with the question, what is BEAM-Therapeutics? Well, BEAM-Therapeutics is a company that is focused on base editing, which is a precision gene editing method that doesn't cut both DNA strands like CRISPR-Cas9, but instead it allows single letter changes, ideal for fixing point mutations, which happen to make up a large portion of human genetic disease. BEAM calls this genetic surgery without the scanning. Their vision is to build a pipeline of curative, once-and-done therapies using this precise, programmable editing tool. So the next question is, what makes base editing different from CRISPR-Cas9? CRISPR uses Cas9 to cut both strands of DNA, which introduces a double-strand break. While that's effective, it can trigger unwanted repair mutations. It could also cause large deletions of DNA or even chromosomal rearrangements, because when a double-strand cut is done on DNA, DNA attempts to repair itself, and that's when these things happen. Base editing avoids all of these. BEAM's base editors are engineered proteins that combine a deaminase enzyme, which chemically changes one DNA letter into another. It also has a Cas9 variant, which positions the editor at the specific DNA site using a guide RNA. And sometimes they use a third optional component, which is called glycosylases, which help clean up the DNA base that is being removed. Now, I have a diagram here that shows how base editor works. Please have a look at it while I explain it to you in the background. So the base editing chemically converts one base like A or adenine into another such as G or guanine without cutting the DNA strand. And this allows transition and transversions, conversion between A, T, G, and C bases, as you can see in the far right side of the screen. Unlike standard CRISPR, there is no double strand break, so the risk of unintended edits or large deletions is significantly lower. This process enables BEAM to precisely fix point mutations, which cause many monogenic diseases. BEAM has developed CBE, that is cytosine base editors, and ABE, that is adenine base editors, in-house, along with a custom lipid nanoparticle delivery system, or LNP. And this platform is now in human trials. This is a platform with modular potential, and it's why investors and partners like Pfizer are paying special attention to BEAM-Therapeutics. Having said that, now let's look at their most advanced programs. The first one is BEAM-101, ex vivo therapy for sickle cell disease, which edits HbF promoting elements. And in my opinion, if approved, this will be the best gene editing therapy for sickle cell disease. It's in phase 1/2 Beacon trial, updated data to be presented at EHA in June 2025. It's targeting 30 patients to be enrolled by mid-2025. And the next therapy we are looking at is BEAM-302. It's an in vivo LNP delivered base editor for alpha-1 antitrypsin deficiency, or AATD. It recently cleared the IND in the US. Early data shows dose-dependent correction of E342K mutation. The dosing of the fourth cohort with 75 mg is underway right now. Full data is expected in the second half of this year. Next, we have BEAM-301, which is also an in vivo therapy which corrects R83C mutation in glycogen storage disease. The first patient was dosed in Q1 of this year. It's in early stage, but it's very important as BEAM's second in vivo clinical program. And just as I was finishing this section, I got a prompt on my screen and I went and investigated it, and I've got some breaking news for you. Beam Therapeutics has just announced that the US FDA has granted RMAT designation to BEAM-302, which is an in vivo base editing therapy for alpha-1 antitrypsin deficiency, or AATD. Now let's take a moment to understand how big this is for Beam Therapeutics. RMAT, or Regenerative Medicine Advanced Therapy designation, is FDA's fast-track designation for regenerative products. It's only given when early data shows that a therapy could significantly improve serious or life-threatening conditions. BEAM-302 was already a standout because it uses LNP to deliver base editors directly to liver to correct the E342K mutation in AATD patients. Now, with RMAT status, Beam will get closer guidance from FDA, including a rolling BLA potentially, priority meetings, and potentially accelerated reviews. According to Beam's president, this confirms that BEAM-302 has the potential to be a one-time curative treatment for a disease with very few good options today. And I think this also reduces the risk for Beam because they'll be doing a rolling BLA most probably. In short, this is the first major regulatory milestone for Beam's in vivo platform, and it's a strong validation of both the science of base editing as well as the strategy. Now let's look at how Beam is building out its partnership in order to support this kind of a clinical push. I guess that most of you by now know that Beam is partnered with Pfizer for undisclosed programs which are in vivo using LNP platform. And it's also partnered with Apellis for autoimmune disorders and FcRn and related targets. I think these give Beam early validation of their technology and funding, while Beam focuses internally on its wholly owned hematology and liver-targeted diseases. Now let's talk about their financials. As of Q1 2025, Beam Therapeutics has cash and marketable securities of approximately around $1.2 billion. And according to their Q1 report for 2025, the company says that cash runway can last through 2028. The net loss was $109 million for the quarter, mostly from R&D. This is a pre-revenue R&D platform company, but one with a diversified approach and sufficient cash to fund major readouts. The recent $500 million direct offering was a strong show of investor confidence, in my opinion, especially after their BEAM-302 early data. Now let's talk about the key milestone to watch for the next 6 to 12 months for Beam Therapeutics. I would start off with BEAM-302, for which we are looking for more safety or efficacy data at a medical conference in the second half of this year. Then we look at BEAM-101, where updated results at EHA is expected around June 2025, which is almost next month. For BEAM-301, we anticipate early safety data for ongoing patient dosing in late 2025. For ESCAPE platform, for the two therapies, BEAM-1003 and 104, we know that healthy volunteer trial for monoclonal antibody is expected to begin later on in this year. So I think we can safely expect many catalysts during the rest of the year for Beam Therapeutics, and that could help or hinder the stock. So retail investors should keep that in mind. Now let's look at the risks and unknowns. As promising as this platform is, retail investors should be aware that there are no approved product yet using Beam's base editing. And Beam is still proving safety of in vivo delivery at higher dosages. Off-target edits, immune reactions to LNPs, and scaling are potential hurdles that we can look forward to. And still, if any of these assets that we discussed succeed, the upside potential is significant. So friends, that brings me to an end of this review. If you found this video helpful, please like and share it with others who would like to follow genomic investing. We do deep dive in this channel every week on companies leading in gene editing, sequencing, and biotech innovation. So if you subscribe and turn on notifications, you won't miss what's happening next. Do you think base editing can outpace CRISPR? That is my question to you. And also, do you think Beam Therapeutics will be the company that leads the way for base editing? Let me know in the comments. Until next time, stay curious and stay invested. Bye for now.